Well, we can't say for sure what the significance of OPN in breast milk is, but we have done studies where we have used the cow's milk corresponding OPN, the bovine OPN, and we have done a clinical trial. And what we saw in that clinical trial is that we changed the immune profile of those infants that got formula with OPN and made them much more similar to breastfed infants, while those that received the formula without OPN were quite different. And different in that sense is that the immune system was different. They had more inflammatory cytokines, they had less anti-inflammatory cytokines, and the question is what it means, but we saw that they had less illness. They had less fever when they got formula with OPN, and they were in that sense similar to breastfed infants, because we know that breastfed infants in general have fewer infections than formula-fed infants. So we were able to reduce the gap between formula-fed infants and breastfed infants when it comes to infection and illness, and it's most likely mediated via the immune system. What I talked about in my presentation today was that we were able to find higher levels of human osteopontin in the infants that were fed formula with the bovine osteopontin, suggesting that if you put OPN into formula, you will evoke a response in the formula-fed infants so they make more of the human osteopontin, which then can have systemic effects. What I talked about earlier with the reduced infections most likely is an effect of OPN on the gut, on the small intestine. So it's a local effect in the intestine. But now, since we have detected OPN being increased in the serum, it can then have effects on different target organs, for example, the brain. And the reason we're interested in the brain is that we have used a mouse model where the mothers are so-called knockout mice, and they produce normal milk with the exception that it doesn't contain any OPN. And then we compare that with mice that have normal levels of OPN. And then they all feed normal mouse pups. And what we see is that the mice that got milk without osteopontin, totally without osteopontin, otherwise normal milk, they had impairment in cognitive function later in life. And we were able to correlate that at the molecular level with changes in expression of proteins in the brain. So we can, in a mouse model, we can explore by looking at specific target tissues, which we cannot do in human infants. So it's not evidence that the OPN in human infants will have an effect on brain, but it's strongly suggestive, and it gives us ideas for future clinical studies to not just look at infections as we did, and immune system, but also look at learning and behavior and IQ. My research group has looked at the composition of breast milk. We have looked at various compartments of breast milk. We have looked at the whey proteins, the caseins, and when looking at the proteins, there is also a group of proteins that is associated with the milk fat globule membrane proteins. And when isolating those, we found that they are very specific, and many of them have biological activities in the laboratory. Some of them are anti-infectious, some of them facilitate nutrient uptake, etc. And what we found was that infant formula does not contain any MFGM, because since the 1960s or 1970s, all the milk fat was taken out of infant formula. Reason being the milk fat being high in cholesterol and being high in saturated fatty acids, And at that time, those were considered really bad, and then we should not expose infants to them because adults should avoid them. So the milk fat was thrown out, and instead vegetable oils were blended. So you got a fatty acid mixture, which was similar to that of breast milk, which is good, but the whole MFGM fraction was missing in infant formula. And I was collaborating with a dairy company in Denmark, and they were able to isolate the MFGM from cow's milk. And we did the first study together in Peru, where we gave infants between 6 and 12 months of age MFGM daily in their complementary food, because we wanted to make sure, even if it was a milk fraction, we wanted to make sure that it was safe, and we didn't want to expose too small infants to this fraction since it had not been given to infants before. And what we found was we looked at infection, and that's what we did the study in Peru. Of course, they have a lot of infections during the first year of life. And we found a significant reduction in diarrhea, in particularly bloody diarrhea. So MFGM added to the diet of older infants could reduce infections. So then we moved on, and we did a study together with colleagues in Sweden, where we added the MFGM to infant formula now. So this is the younger age bracket, between 1 and 6 months of age. And then we followed them up to 12 months of age. So we had a breastfed control group, and then we had regular formula, and we had the experimental formula with MFGM added. And what we found in that study too was that we had significantly less infections in the infants that got the formula with the MFGM. And they became similar to the breastfed infants, while those that were fed the regular formula had more infections, which is well known that formula-fed infants have more infections than breastfed infants. And in particular, we found a significant reduction in ear infections or otitis media. And that is well known that breastfed infants get much fewer ear infections than formula-fed infants. And we were able to mimic the breastfed infants in that sense by adding MFGM. But we also looked at cognitive development, because the MFGM doesn't only contain proteins, it also contains very important lipid building blocks for the brain and some carbohydrates, particularly sialic acid. So we wanted to explore if we could see an effect on cognitive development at 12 months of age. And it's well known that at 12 months of age, formula-fed infants do not perform as well as breastfed infants. And in this study, which was a relatively large study with 80 infants in each group, we found that addition of MFGM to formula made their cognitive development similar to that of breastfed infants, while those that received the standard formula did not have the same cognitive development. So we had two major outcomes of the study: fewer infections, particularly ear infections, and we had improved cognitive development. And these have been two of the major differences between formula-fed and breastfed infants. So we were quite happy with the outcome of that clinical study. And since that time, several companies have now added milk fat globule membrane fractions to their formulas. I think one of the most exciting findings is that the last several years it has been found that breast milk contains small pieces of RNA, so-called microRNAs. They are a very specific size, about 20 to 24 nucleotides, and they are there in very specific composition. And not only that, when we have exposed them to conditions that you have in the gastrointestinal system, that is, you expose them to the high acid in the stomach, enzymes, the enzymes of the pancreatic fluid, etc. Everything that breast milk will be exposed to on its way down into the intestine of infants, these microRNAs, not only do they survive digestion, but they are taken up by human intestinal cells in culture. So we expose these cells that we have in the laboratory that are from human infants, and they take up these microRNAs. And microRNAs are regulators of gene expression. So they are very potent molecules. They can affect the expression of a lot of other components in the body, proteins. First, we thought that most likely it will be effective in the small intestine. It could affect the immune system, for example. But then we have also found, and others have found, that microRNAs are actually also in the serum of the infants. And therefore, the possibility exists that they can go to other target tissues in the body, such as the liver, the kidney, the brain, etc. So these microRNAs can have potentially very important effects on the development of the breastfed infant. So what we have found also is that it's not just these random small pieces of RNA. When we compare our published results from women in California, there are the same 15 top microRNAs, the most abundant, the most common ones, are the same in California as they are in China, as they are in Australia, showing that the maternal body, the mammary gland, has decided that these are the ones we are going to make. And then you start as a scientist to ask, well, if the mammary gland has put high priority on making these precise microRNAs, what is the purpose of that? So that's about where we are. There are specific microRNAs. They will survive. They will go into the body. What will they do? And that is, I think, a field that is going to expand and move forward. And I think we need a lot of basic research first, because these are very potent molecules, and even if they can be synthesized and potentially added to formula, it would be way too early to do experiments in human infants before we know much more about their action, the dosage, how much can be given, and if they should be given. But I think it's an angle of research that is very important and that should be pursued, and there are several research groups that are looking at this.